Introduction to clinical laboratory. Clinical laboratory testing is fundamental to modern healthcare, playing a crucial role in the detection, diagnosis, and treatment of various diseases. These tests provide objective data that guides medical decisions, often impacting up to 70% of all medical diagnoses. After collecting and examining a specimen, laboratory professionals meticulously analyze the samples and communicate the results to physicians or other primary care providers. This critical information enables healthcare teams to make informed decisions regarding patient care and treatment plans. The demand for skilled laboratory professionals is consistently high, with employment in the profession expected to grow faster than the average for all occupations. This growth reflects the increasing complexity of diagnostic testing and the aging global population. Early professional organizations. The foundation for credentialing and professional standards in clinical laboratory science was laid in the early 20th century. These early efforts were crucial for establishing the credibility and expertise of laboratory professionals. In 1928, the American Society for Clinical Pathology, ASCP, established the Board of Registry, BOR, to certify laboratory professionals. Individuals who successfully passed the BOR's registry exam were initially designated as medical technologists, identified by the acronym MT-ASCP. This marked a significant step towards formalizing the profession. Further professional organization occurred in 1933 with the formation of the American Society of Clinical Laboratory Technicians, ASCLT. This organization has since evolved and is now known as the American Society for Clinical Laboratory Science, ASCLS, continuing its mission to advocate for and advance the profession. Evolving professional roles. The clinical laboratory profession saw significant expansion and specialization during the 1960s. New categories of laboratory professionals emerged, complementing the work of generalist medical technologists in performing daily clinical laboratory tasks. A key development was the introduction of the medical laboratory technician (MLT) role, typically requiring a 2-year associate's degree program. This created a tiered system, allowing for a broader range of expertise and entry points into the field. Specialist categories also proliferated, including those in chemistry, microbiology, hematology, and blood banking. Further specializations evolved in areas like cytotechnology, histotechnology, laboratory safety, and molecular pathology, molecular biology, reflecting the increasing complexity of diagnostic science. For example, a cytotechnologist plays a crucial role in cancer detection by examining cells for abnormalities. Licensure and professional recognition. Beyond initial certification, many states and U.S. territories require licensure for clinical laboratory professionals to ensure public safety and maintain high standards of practice. This legal requirement adds another layer of accountability. States and territories currently requiring licensure include California, Florida, Georgia, Hawaii, Louisiana, Montana, Nevada, New York, North Dakota, Rhode Island, Tennessee, and West Virginia, as well as Guam, Northern Mariana Islands, and Puerto Rico. The specific requirements for licensure can vary significantly by state and the particular laboratory specialty. In 2009, a significant professional recognition occurred when generalist medical technologists became known as medical laboratory scientists, MLSs. The technician-level designation continued as medical laboratory technicians, MLTs, with appropriate credentialing being MLS ASCP and MLT ASCP. Maintaining these certifications often requires ongoing continuing education, ensuring professionals stay current with advancements and best practices. Advanced practice, doctorate in clinical laboratory science. The profession continued to advance with the approval of the post-baccalaureate degree, the Doctorate in Clinical Laboratory Science, DCLS, in the United States in 2012. This marked a new era for advanced practice within the field. The DCLS credential represents a terminal advanced practice degree, extending beyond the entry-level generalist role. It prepares professionals for leadership and specialized roles that require a deeper understanding of laboratory medicine and its integration into patient care. The National Accrediting Agency for Clinical Laboratory Sciences, NAACLS, categorizes the responsibilities of a DCLS into 5 key areas: patient care management, education, research applications, healthcare policy development, and healthcare services delivery and access. This broad scope allows DCLS professionals to significantly impact healthcare at multiple levels, from direct patient consultation to shaping national health policies. Core clinical laboratory functions. A primary function of the clinical laboratory is to educate physicians and other healthcare providers. This ensures that the information derived from reported test results is used appropriately and effectively in patient management. When tests are ordered, the clinical laboratory assumes a crucial role of leadership and education. This involves assisting the physician in making decisions that serve the best interest of the patient, improving the clinical decision-making process, and carefully considering the associated costs. While a vast array of diagnostic tests are available, typically only a small percentage are routinely ordered. However, these selected tests are vital as they significantly improve clinical decision-making, leading to more accurate diagnoses and targeted treatments. For instance, a complete blood count, CBC, is one of the most frequently ordered tests, providing essential information about a patient's overall health. Laboratory leadership and staffing. The leadership of a clinical laboratory is typically overseen by a pathologist or a PhD-level scientist. This director is responsible for the overall scientific and medical integrity of the laboratory operations. Clinical laboratory personnel include a hierarchy of roles, starting with laboratory supervisors or managers who are responsible for the technical aspects of managing the laboratory. Additionally, a business manager may handle the administrative details, ensuring smooth financial and operational functions. The business manager's role is crucial for ensuring that all federal, state, and local regulatory mandates are meticulously followed by the laboratory. The core workforce comprises technologists, technicians, and specialists who perform the hands-on testing and analysis. This collaborative structure ensures both scientific excellence and operational compliance. Duties of laboratory professionals. The responsibilities of medical laboratory scientists, MLSs, and medical laboratory technicians, MLTs, are diverse and can vary based on the laboratory's size and specialization. Their duties may include performing a wide range of laboratory assays, supervising other staff, engaging in teaching, or conducting research. Due to their in-depth knowledge of technical aspects, principles of methodology, and the instrumentation used for various laboratory assays, laboratory professionals are uniquely positioned to correlate and interpret complex data. This analytical skill is critical for accurate diagnosis and patient care. While MLSs and MLTs are capable of collecting blood specimens or processing them, phlebotomists are typically responsible for blood specimen collection in larger hospitals. This specialization allows for greater efficiency and expertise in venipuncture, ensuring patient comfort and proper sample integrity. CLIA 88, ensuring quality lab results. The Clinical Laboratory Improvement Amendments, CLIA, of 1988 were enacted with the primary goal of ensuring high quality and accuracy of laboratory results across the United States. This federal regulation applies to all facilities that perform laboratory testing on human specimens for health assessment or for the diagnosis, prevention, or treatment of disease. CLIA establishes specific requirements for laboratory personnel, defining their responsibilities, educational qualifications, necessary training, and experience based on the complexity of the tests they perform. This tiered approach ensures that staff are adequately prepared for their roles. Notably, there are no CLIA regulations for testing personnel who work at sites performing only waived tests or provider-performed microscopy (PPM) testing, as these are considered lower risk. However, requirements become significantly more stringent for those performing tests categorized as highly complex, reflecting the greater potential for patient harm if errors occur. This regulatory framework is critical for maintaining public trust in diagnostic testing. CLIA test classification criteria. CLIA categorizes general laboratory testing into three main levels: waived tests, moderately complex tests, and highly complex tests. This classification system is designed to match the level of regulatory oversight with the potential risk associated with each test. The criteria used for classifying tests are comprehensive and focus on patient safety and result reliability. These include the potential risk of harm to the patient if the test is performed incorrectly or if an erroneous result is reported. Further criteria involve the type of testing method used, the degree of independent judgment and interpretation required by the testing personnel, and the availability of the test for home use. For example, a test requiring highly specialized equipment and extensive interpretation by a trained professional would be classified as highly complex, demanding stringent oversight. Waived and provider-performed microscopy tests. Waived tests represent the simplest category of laboratory tests under CLIA. These tests are cleared by the U.S. Food and Drug Administration, FDA, for home use, making them accessible for individuals to perform outside of a traditional laboratory setting. The methodologies used for waived tests are straightforward and are considered unlikely to cause erroneous results or pose any reasonable risk of harm to the patient if performed incorrectly. Common examples include dipstick urinalysis, which checks for various substances in urine, and blood glucose monitoring, essential for diabetes management. Provider-performed microscopy (PPM) tests are another specific category. These tests must be personally performed by a qualified practitioner, such as a physician, a mid-level practitioner under physician supervision, or a dentist. PPM tests are categorized as moderately complex, requiring a certain level of professional expertise. PPM test characteristics and limitations. For a test to be classified as provider-performed microscopy (PPM), the primary instrument used for its performance must be a microscope, specifically limited to bright field or phase contrast microscopy. This restriction ensures that the tests remain within a defined scope of complexity. A critical characteristic of PPM specimens is that they are often labile, meaning they are unstable and can degrade quickly. This necessitates immediate examination after collection, as control materials for these types of specimens are typically not available. Furthermore, specimen handling for PPM tests is usually limited, reinforcing the need for prompt analysis. These characteristics collectively define PPM tests as moderately complex, requiring the direct involvement and expertise of a qualified practitioner to ensure accurate and timely results. Laboratory organizational models. The organizational structure of a clinical laboratory is highly adaptable, depending on its size, the volume and type of tests performed, and the available facilities. This flexibility allows laboratories to optimize their operations. Larger laboratories often adopt a departmentalized structure where separate areas are designated for various specialized divisions. These can include cytogenetics, toxicology, flow cytometry, and other specialized divisions like molecular diagnostics, each focusing on specific analytical techniques and disease areas. A growing trend is the adoption of a more open design or a core laboratory model. In this setup, disciplines such as hematology, urinalysis, hemostasis coagulation, and clinical chemistry share a common workspace. Cross-training of staff is particularly important in a core laboratory model, enhancing efficiency and flexibility by allowing personnel to work across multiple areas. Traditional and core lab disciplines. Traditionally, a working clinical laboratory is organized into several major scientific disciplines, each focusing on a specific area of laboratory medicine. These disciplines ensure comprehensive diagnostic capabilities. These traditional disciplines include blood banking, transfusion medicine, clinical chemistry, which may encompass toxicology, flow cytometry, hematology and hemostasis, immunology and serology, microbiology, urinalysis. Each specialty department focuses on a different area, providing in-depth analysis for specific types of samples or conditions. The core laboratory configuration, a modern approach, combines routine hematology, hemostasis, and blood coagulation, and clinical chemistry into a single, integrated workspace. This consolidation aims to improve turnaround times and resource utilization for high-volume tests, streamlining operations and enhancing overall efficiency. Molecular diagnostics and future directions. Many medium to large-size laboratories have strategically developed central testing areas equipped with clusters of instruments specifically designed to handle high volumes of test samples. This centralization optimizes workflow and resource allocation for routine and specialized tests. Expanded directions in laboratory testing increasingly include molecular diagnostics, which represents a significant application of biotechnology. This field applies the fundamental principles of basic molecular biology to the study and diagnosis of human diseases. Molecular diagnostics provides real-time information crucial for advanced applications such as gene therapy, genetic screening, stem cell research, cloning, and cell culture. For example, PCR, polymerase chain reaction testing, a molecular diagnostic technique, is widely used to detect infectious diseases like COVID-19 and genetic mutations, demonstrating its immediate impact on patient care and public health. Healthcare organization structure. Modern healthcare organizations exhibit diverse configurations, which are influenced by factors such as their geographic region, market dynamics, patient mix, overall size, and affiliations. This variability leads to a wide spectrum of operational models. The size of these organizations can range dramatically, from very large tertiary care level teaching hospitals that serve complex cases and train future medical professionals, to smaller community hospitals, freestanding specialty clinics, or even basic phlebotomy drawing stations. A common organizational structure for a hospital typically includes a chief executive officer, CEO, and a board of trustees, who are responsible for setting policy and guiding the overall direction of the organization. The chief operating officer, COO, then takes charge of implementing these policies and managing the daily activities, ensuring smooth and efficient operations. Healthcare leadership roles. Beyond the CEO and COO, other high-level positions are integral to healthcare organizations, varying based on the institution's size and specific needs. These can include the Chief Financial Officer, CFO, Chief Information Officer, CIO, and Chief Technology Officer, CTO, each managing critical aspects of the organization. A variable number of vice presidents typically oversee various departments, with several reporting directly to them. This structure allows for specialized management across different operational areas. Common VP roles include vice presidents of nursing, clinical services, general services, and human resources. Notably, the VP of Clinical Services holds a broad oversight, managing not only the clinical laboratory, but also other vital departments such as radiology and pharmacy, ensuring integrated and efficient patient care services. Primary laboratory accrediting organizations. To ensure quality and compliance, clinical laboratories are subject to accreditation by several primary organizations. These bodies play a crucial role in maintaining high standards across the industry. One such organization is the Commission on Office Laboratory Accreditation, COLA, which focuses on accrediting physician office laboratories and other smaller facilities. COLA's mission is to promote excellence in laboratory medicine through a peer-based accreditation process. The College of American Pathologists, CAP, offers an internationally recognized accreditation program. CAP distinguishes itself by utilizing teams of practicing laboratory professionals as inspectors, ensuring that evaluations are conducted by experts with real-world experience. This peer review model is highly respected for its rigor and relevance. Another key accrediting body is the Joint Commission, TJC, which has been evaluating and accrediting hospital laboratory services since 1979. TJC expanded its scope to include freestanding laboratories in 1995, providing comprehensive oversight for a wide range of facilities. TJC's accreditation is often a prerequisite for Medicare and Medicaid reimbursement. Other specialty accrediting organizations. In addition to the primary accrediting bodies, several other specialty organizations provide accreditation for specific types of facilities or services within laboratory medicine. These specialized accreditations ensure expertise and quality in niche areas. These organizations include the American Association of Blood Banks, AABB, the American Society of Histocompatibility and Immunogenetics, ASHI, the American Osteopathic Association, AOA. Each of these bodies focuses on particular aspects of laboratory practice, such as blood product safety, tissue typing for transplantation, or osteopathic medical facility standards. The existence of these specialty organizations highlights the vast and intricate nature of laboratory medicine, where different areas require tailored oversight to meet unique challenges and ensure the highest level of patient care in specialized fields. External government regulation and safety beyond CLIA 88. Clinical laboratories are subject to a comprehensive array of other state and federal regulations. These regulations govern critical aspects, such as chemical waste disposal, the safe use of hazardous chemicals, and overall laboratory safety protocols. A key area of regulation involves the handling of biohazardous materials and the application of standard precautions, which are universal infection control practices. External controls also include standards mandated by public health laws and reporting requirements through the Centers for Disease Control and Prevention, CDC, along with certification and licensure requirements issued by the U.S. Food and Drug Administration, FDA. State regulations are imposed by Medicaid agencies, state environmental laws, and state public health and licensure laws, ensuring localized compliance. Furthermore, local regulations, determined by building codes and fire prevention codes, add another layer of safety oversight. This multi-layered regulatory environment ensures a safe and compliant operational setting for all laboratories. Alternate sites of testing. The landscape of laboratory testing has evolved to include various alternate sites, moving beyond the traditional central laboratory model. This decentralization of laboratory testing offers flexibility and quicker access to results. Point-of-care testing, POCT, is a significant aspect of this decentralization, referring to diagnostic testing performed near or at the site of patient care. POCT devices are often portable and user-friendly, allowing for rapid results in clinics, emergency rooms, or even at a patient's bedside, which can significantly impact immediate treatment decisions. Reference laboratories are specialized facilities that perform highly complex, esoteric, or low-volume tests that may not be feasible for smaller or general laboratories to conduct in-house. These labs serve as a crucial resource, offering advanced diagnostics and specialized expertise. Physician office laboratories, POLs, are another alternate site where tests are performed directly within a physician's practice. POLs typically conduct waived or moderately complex tests, providing convenience for both patients and practitioners by integrating testing directly into the clinical visit. Patient rights, informed consent, and confidentiality. In the realm of medical legal issues, informed consent is paramount. It means that a patient is fully aware of, understands, and explicitly agrees to the nature of the testing to be done, including the procedures involved and how the results will be reported. This ensures patient autonomy and participation in their healthcare decisions. Confidentiality is another cornerstone of patient rights, legally reinforced by the Health Insurance Portability and Accountability Act, HIPAA, enacted in 1996. HIPAA established national standards for the protection of sensitive patient health information. Under HIPAA, any results obtained from patient specimens and any information about the patient must be kept strictly confidential. This legal mandate protects patient privacy and fosters trust between patients and healthcare providers, ensuring that personal health data is secure from unauthorized access or disclosure. New patient access and chain of custody. Recent advancements in patient access regulations have empowered individuals with greater control over their health information. A new final rule by the Centers for Medicare and Medicaid Services, CMS, now grants patients direct access to their laboratory results, promoting transparency. It is important to note that this new rule does not require laboratories to interpret test results for patients. While patients have direct access, laboratories may continue to refer patients with questions about their test results back to their ordering or treating healthcare providers, ensuring appropriate medical context and advice. When specimens are involved in possible medico-legal situations, stringent specimen handling policies are required, known as maintaining the chain of custody. For evidence to be admissible in legal proceedings, every step of the analysis must be meticulously documented, from the moment the specimen is collected and transported to the laboratory, through the analysis itself, and finally to the reporting of the results. Any break in this chain can compromise the integrity of the evidence. Other legal considerations and medical ethics. Beyond specific regulations, healthcare organizations and their employees are legally obliged to provide an acceptable standard of care. This is defined as the degree of care that a reasonable person would take to prevent an injury to another, encompassing all aspects of patient interaction and service delivery. Medical ethics form the bedrock of professional conduct, encompassing both personal and professional principles. Personal ethics are based on an individual's or group's values, ideals, and customs, which are held in high regard and guide moral behavior. Professional ethics, on the other hand, encompass the specific principles of conduct expected within a particular profession. The American Society for Clinical Laboratory Science, ASCLS, strongly endorses a professional code of ethics, which outlines the responsibilities of all laboratory professionals for proper conduct toward the patient, colleagues, and the profession, as well as society at large. In addition, ASCLS has a pledge to the profession reinforcing these commitments.